Hello, everyone. My name is Kelly Hall. I'm a marketing coordinator for ALS North America. I want to thank you all for joining us today for our Webinar Wednesdays series. Our presenter today is Jordan DeSantis. Jordan received her bachelor and master's degree from Arizona State University. She has been overseeing clinical claims substantiation testing in the sunscreen testing department for the past 4 years and has been working for BioScreen Testing Services for the past 8 years. Now Jordan serves as the clinical manager at our ALS BioScreen Testing Services laboratory in Phoenix, Arizona. Today, she will be discussing the various testing methodologies required for sunscreen labeling, including SPF label claims, UVA protection, and safety testing. During this presentation, you may use the chat box to ask a question or raise the hand feature to ask questions. We will answer all questions at the end of her presentation. Jordan, thank you for taking your time to deliver this webinar today. I will go ahead and turn it over to you.
Great, thanks Kelly, I appreciate it. Okay, so today I'm just going to kind of discuss a little bit about the sun protection factor or SPF testing that we do here at BioScreen. I'm going to cover, generally speaking, the testing methodologies that we have and perform, as well as some additional changes that may be coming up in the testing regulations. So BioScreen offers sun protection factor or SPF testing to comply with the FDA and international testing regulations. The most commonly used are the FDA and the International Standard 24444 for SPF label claim testing. We also perform the other international testings specific to in vitro testing and UVA protection. Additionally, the CALIPA, Australia, New Zealand, and JCIA methods that are specific to, are more specific to specific countries. The testing equipment that we use here at BioScreen is calibrated at least annually by an independent facility to irradiance standards traceable to NIST to certify the spectral compliance with the testing standards. So this company comes in, checks our instruments, and they make sure that the instrument's output or the UV complies with the testing regulations, be it FDA, the international testing standards. We additionally also check our instruments' light sources. They're checked on a daily basis for intensity of the UV exposure and to ensure that the UV beams are uniform. BioScreen is currently equipped with 6 single-port solar simulators and 4 multi-port solar simulators for SPF testing. So I'm going to touch on a little bit on the actual equipment itself to kind of help gather an understanding of the test further. We mentioned we use a single port and a multi-port solar simulator. This single port solar simulator utilizes a 150 watt ozone-free short arc xenon lamp. It has interchangeable sensors, so we can detect the UV spectrum of UVA plus UVB, or we can filter it and detect only UVA spectrum. It utilizes an automatic dose controller so that we can continuously monitor our UV output to ensure that we're providing accurate UV doses. The single port, based on its name, only allows for one controlled UV dose to be given at a specific time. You'll also see that it's fitted with what we call a patient stop. So it's that little arm that projects out from the solar simulator. This patient stop is used as essentially a backrest for participants. This will ensure that our participants are all at an equal distance from the light source, and also ensures lack of them moving during the course of the test. It's essentially used to ensure repeatability. When using a single port solar simulator, our subjects are always in a seated position for the duration of the testing. We also use what's called a multi-port. The multi-port utilizes a 300 watt ozone-free short arc xenon lamp, so it's a little bit more powerful. The reason for this is that instead of one particular output for UV, it's fitted with 6 individual square outputs. Because it will be irradiating 6 outputs versus 1, we need that extra power. Each output can be controlled individually to provide a specific dose or to be turned off completely. So although there's 6 output and what we'll call ports, you can use all 6, or you can use 1 if that's what you choose. It's fitted with an articulated arm that can be adjusted for subject positioning. The subject's position for this particular test is that they're always going to be in a prone position or laying down. However, we can articulate the arm to accommodate for the curvature of the test site. The multiport allows for selection of either UVA plus B or UVA only, similar to the single port. Again, it also has interchangeable sensors, so we can detect those solar spectrums specifically. It uses the same automatic dose controller so that we can monitor the UV output and provide accurate dose amounts. And as mentioned, the subject is in a prone or lying down position for the duration of the test. Subject selection, generally speaking, the subject selection between the FDA and the International Testing Regulation are fairly similar. And this is specific to SPF determination only, so a UVA plus UVB test. Subjects must either be Fitzpatrick skin type 1, 2, or 3. Fitzpatrick skin type is determined based on the first 30 to 45 minutes of sun exposure following a winter season of no sun exposure. It ranges from a Fitzpatrick 1 to 6. Fitzpatrick 1 is defined as somebody who always burns easily and never tans. So they're the most sensitive to sunlight. That will increase up to a Fitzpatrick 6. This person usually never burns. They're deeply pigmented. They're considered insensitive to sunlight. The international standard not only uses Fitzpatrick currently as a method to determine skin type, but they also have what's called an ITA, or an individual typology angle. This is essentially another method for evaluating skin color. The ITA utilizes a chromometer. This device measures the skin color on a participant's back. The international standard uses this method in addition to Fitzpatrick because the traditional Fitzpatrick phototype classification is based on a subject's experience of their own sensitivity to sun exposure and their ability to tan, which can be subjective. This could lead to inappropriate UV doses. The ITA uses a quantitative analysis and classifies skin tone as either very light, light, intermediate, tan, matte, or brown. So it's a more definitive approach to determining skin type. The subjects that we ask to participate in studies should not be taking any topical or systemic medication that's known to alter responses to UV. And that could be either to worsen or diminish the responses. The test itself for SPF determination is looking for an erythemal or a reddening response. If somebody's taking a medication that would increase the reddening response or diminish the reddening response, will have an impact on the test itself. We're looking for individuals who have no history of sensitivities to topical products. Obviously, we don't want to put a product on somebody who has a known sensitivity because perhaps they'll break out in a rash. So not something that we want to include in our test. Additionally, we're looking for individuals who have been adequately evaluated for uniform skin color. That means that there is no excessive pigmentation, nevi, moles, sunburns, excessive hair, any sort of disfigurations like scars on the back, which is our test area. These kind of things, again, would not allow us to read a proper erythemal response following UV exposure. The test site itself is always on the back. It's to the left and right of the midline and just below the scapular region, or the shoulder blades. The first step in testing for SPF determination is to determine what we call a subject's unprotected MED, or their minimal erythemal dose. The minimal erythemal dose is defined as the area on a subject that produces the first perceptible reddening or erythemal response at the lowest dose possible. With this information, we then determine all of our UV exposures later on in the test when we're physically testing the sunscreen or the test sample. In order to determine a subject's MED, a test area is demarcated on the subject's back. Within that test area, we also demarcate 5 additional subsites. These 5 subsites are areas that are going to be exposed to UV. In the 5 subsites, we'll start with the lowest UV dose, and then we will progressively get higher with UV doses as the subsites go up. Each subsite uses a 25% progression. So the first subsite is your lowest dose. The next subsite will be 25% higher than that. The third, 25% higher than the second, and so on. Following this UV exposure, subjects are dismissed from our facility and are instructed to come back 16 to 24 hours later so that we can take a look at their back and see where we find the minimal erythemal dose. When subjects come back, they're evaluated in the same position in which they were irradiated by a trained grader using sufficient lighting. So if we were using a single port solar simulator, the subject would then again be evaluated in a seated position. Using a multi-port solar simulator, when the subject comes back for evaluation, we'll have them lie down again because that's how they're irradiated. As mentioned, the MED is determined by the subsite where the first perceptible unambiguous erythemal response is noted. So for a quick example, obviously the test area is a little lighter because this person came back almost 24 hours later. However, you could see the delineation of the test site. You'll see it's in a rectangular shape. And then within that area, we had 5 circular subsites drawn. So the lowest UV dose was given at the very first circular subsite. You can see all the way to the left of your screen. And then UV doses that were progressively higher throughout the remaining test sites were given. When they came back, we're looking for the first unambiguous perceptible erythemal response. This we can see is at the 3rd test site. You could see that the single port solar simulator, which is what was used for this test, provides a circular UV exposure, so you can see it has some nice defined borders. There is an erythemal response that covers that entire circle. And then you can also see that the erythemal responses continue to the right of that exposure site, which would be expected because to the right of that site, we gave higher UV doses. So we would expect that that erythemal response would continue. So on this particular subject, we would determine that the dose that was given at that 3rd site would be considered the subject's MED. Once the MED is determined, we will then demarcate additional test sites similar to the first. Again, they'll have 5 circular subsites within. These test sites will be specific to the sunscreen product that we're testing. We'll also delineate another site for a standard sunscreen product. The standard sunscreen product is used as a control in all of our SPF testing, and this is to ensure accuracy of our results. The standard sunscreen product comes from an outside facility that had been manufactured and previously tested to ensure that it hits a specific SPF value. So it is a known SPF value. And then we'll also delineate another test site to use as another MED determination or an untreated site. We do this because we use it as a verification of our results. So essentially, we're confirming that the original MED that we provided is still accurate and valid. Test products are then applied to their respective test sites at a dose of 2 milligrams per centimeter squared. All products are shaken or stirred to ensure uniform dispersion. There are some sunscreen products out there that the active ingredients may separate at some point. So this ensures that all ingredients are as uniform as possible within the sample itself prior to us testing it. We use a weight by loss method to ensure proper application amount. So essentially, we weigh all of the products used in the application itself, so a finger caught or a glove along with the syringe. We add the product to the syringe, weigh it again, and then we'll apply the product to the test area and do a final weight to ensure that we applied 2 milligrams per centimeter squared to the site itself. Following application, we check the uniformity of the test product to ensure that there is a nice, even coverage of the test product throughout the entire test site, and we do that by using a Woods lamp. If you're unsure what a Woods lamp is, it's essentially a UV light, and it will show us areas that maybe do not have proper coverage or areas that may be lighter compared to others, indicating that the coverage in that specific area hasn't been as uniform. It allows us to then go ahead and spread the product more evenly so that we have that nice even coverage. Following the application, the products are given time to dry. For the FDA specifically, they require a minimum of 15 minutes. The International Testing Standard requires 15 to 30, so they're a little bit more specific. For liquid type products like lotions, creams, sprays, milks, droplets of the product are just deposited within the test site using the syringe and spread evenly over the test site using light pressure. So you can kind of see in the image below an example of how the product is applied into the entire test site and not necessarily just the little areas that are getting UV exposure. Again, this is to ensure that you're not focusing on those areas and maybe producing some invalid results. As for powder, tapped and spread over the test site. The international standard allows for water to be applied to the test site prior to a powder. It allows for aid in dispersion of the product over the test site. Additionally, it allows for powders to adhere better to the skin. Currently, the FDA does not recognize powders as a suitable dosage form for SPF testing. However, that's up for discussion a little bit further down. Following product application, we provide the UV doses over the applied sunscreen. The middle dose in the series, so the third subsite of the 5, is calculated according to the subject's MED, the previously determined untreated site, multiplied by the expected SPF of the test sample. The remaining doses, both higher and lower, so subsites 4 and 5 would be higher and subsites 2 and 1 lower, are determined by a progression dependent on the expected SPF value of the sample itself. For the FDA, they break it down into 3 progressions. For an expected SPF of less than 8, we use a 25% dose progression, so one dose is 25% higher than the previous. For an expected SPF of 8 to 15, it's a 20% dose progression. And for an expected SPF greater than 15, it's a 15% dose progression. The international standard only breaks it down to 2. It's either less than 25 or greater than 25. For less than 25 expected SPF, we use a 25% dose progression, greater than 25, a 15%. Water resistance testing. So what we just went over previously is what we call a static or a dry determination. That SPF test will give you specifically your SPF label claim. If you wish to determine if your product is water resistant, following product application and drying time, the subjects are placed in a water immersion device, something similar as a pool, whirlpool, hot tub, bathtub, something with water jets that allows for water to flow throughout the immersion device, followed by the UV exposures. So it's essentially one extra step thrown in after product drying and prior to UV exposures. The water resistance tests vary a little bit more than your standard SPF testing between FDA and the International Testing Standard. The FDA allows for either 40 or 80-minute water immersion tests. The FDA only requires a water immersed or a wet determination, so it does not require a dry or just static. The water temperature is 23 to 32 degrees Celsius. They require fresh water, which they define as clean drinking water that meets the FDA standard 40 CFR Part 141. The test itself consists of 20-minute water immersions, followed by 15 minutes rest out of water. This procedure is repeated until you get the amount of time that you're looking for your water immersion test, either 40 or 80 minutes. So for a 40-minute test, it would be a 20-minute immersion, 15-minute rest, 20-minute immersion, 15-minute rest. During the water immersions, they're instructed to perform moderate activity during this immersion, and this is a simulated water activity. So the reason that these water immersion devices have jets or some sort of method of moving the water is because that is what's going to simulate your activity. During the test sites are never toweled, so when the participants are removed from the water, the test sites are left to air dry. Toweling would obviously remove the test product that's currently on their back. And then for the FDA, the label SPF claim is determined from the UV exposures after water immersion. So that is just another way of saying that they have this particular claim has nothing to do with a dry or static determination. It is strictly from a water immersion test. The international testing standard currently does not have a standardized method for water resistance. So in order to do this, the two most widely accepted and used are both the CALIPA and the Australia-New Zealand methods. CALIPA is most widely accepted in the majority of countries. It allows for either a 40-minute or an 80-minute water immersion test, similar to the FDA. However, this test requires both a static and a water immersed wet test. The water temperature for this test is 27 to 31 degrees Celsius. Again, it requires fresh water or clean drinking water. They require a water hardness between 50 to 500 milligrams per liter. Similar to the FDA, it's a 20-minute water immersion followed by a 15-minute rest out of water. And during the water immersion, again, moderate activity is performed using that simulated method. Test sites, again, are never toweled. The product is considered water resistant based on CALIPA standards if the 90% lower confidence interval limit is greater than or equal to 50%, and the 95% confidence interval on the mean static SPF is within plus or minus 17%. So essentially what this is doing is they are comparing the product post-water immersion to the product pre-water immersion. So that is why you have to require both a dry and a wet determination. If the post-water immersion statistical criterion and the pre-water immersion statistical criterion are met, the product can be labeled as water resistant, either 40 or 80 minutes. For Australia and New Zealand, it's one of the more strict testing methods. The Australia-New Zealand method allows for a minimum of 40-minute water immersions, but up to 4 hours. This is dependent on the SPF value, though. Lower SPF values, such as an SPF 15, will only allow up to 40-minute water immersions, whereas the higher SPF values can be immersed up to 4 hours. This test, similar to FDA, only requires a water immersed or a wet determination, so there's no static or dry. The water temperature is 21 to 35 degrees Celsius. pH is 6.8 to 7.2. Again, requires fresh water, clean drinking water. Also requires water hardness levels. The difference in the water immersions is the rest period out of water. So this test requires a 20-minute water immersion followed by a 5-minute rest out of water. Again, it's repeated until we hit the water immersion time per request, which again can be up to 4 hours. The simulated moderate activity is a little bit more specific in the Australia and New Zealand, whereas water is circulated for 16 minutes and then air agitated for 4. Test sites again are never toweled. And then the label claim is determined from the UV exposures after water immersion. So again, this is just saying that there is no comparison to the dry or static test. It is strictly after water immersion. After these tests are complete, either dry or static, and then water immersion, we ask the subjects to come back 16 to 24 hours later, again, and we'll assess for the erythemal responses. The visual grading that's performed is done in a blind manner. So the observer or the visual grader is not the same person who performed the exposures or did the product application. This is to ensure that there's no bias in the grading. Visual grading is done in a room with matte or neutral walls, under a sufficient light source. And then visual graders have an annual eye exam for both visual acuity and color blindness. Of course, we're looking for an erythemal or reddening response, so color blindness could be a problem. The test subsite, again, that provides the first perceptible, unambiguous erythemal response will determine the SPF value of the test product, just similar to the grading of the MED that was described earlier. So I'm going to bring that picture back, and we're going to kind of go through a little bit of an example of the potential responses that could happen during an SPF test. For example, let's say we're targeting an SPF 15. As mentioned earlier, the middle dose that is given, so that third subsite you see, would be the MED, the untreated dose, multiplied by the expected SPF. So if we see that our first perceptible response, erythemal response, is at site 3, our calculations would then tell us at the end of the day that that is an SPF 15 product. So it's meeting exactly what our target SPF was. Now we're going to use our imagination. Let's assume that that third response is not there, but our erythemal responses start at subsite 4. This is now telling us that the SPF is a little bit higher than the target SPF. It is protecting a little bit better than we had targeted. If the SPF showed at subsite 4, we would have an SPF 18. Again, if we had no responses all the way up to subsite 5, it's telling us that the SPF is a 21.6. And then to take it down the other direction, if we had our first erythemal response at subsite 2, we would have a 12.5. And then if we were to have erythemal responses starting at subsite 1 and throughout the rest of the subsites, we would have what's called invalid or rejected results. This is telling us that our SPF is less than or equal to 10.4. Less than or equal to because we don't really know what the SPF value would be as if that were the starting subsite of our erythemal responses. And we'll kind of go into a little bit more about invalid and rejected results. So data is invalidated or rejected for a number of reasons. The exposure series on a subject may fail to elicit an erythemal response on any subsite. So we may not see any erythemal responses whatsoever. We may also see erythemal responses where all subsites in the exposure series show a response. We may see erythemal responses within an exposure series that are randomly absent. So essentially what that would mean is that maybe we see erythemal responses at sites 2 and 3, nothing at 4, and then maybe an erythemal response at 5. So it's inconsistent with the doses that we would give. Because we're starting at the lower dose and progressively getting higher through the subsites, you would assume that once you start seeing an erythemal response, erythemal responses would continue as the doses get higher. Lastly, we would invalidate or reject results if a test subject is non-compliant. Let's say we did our UV exposures and sent our subject home for the day and they went tanning. Obviously, that is additional UV exposure on top of the UV exposure that we just provided. So that's not going to give us accurate or valid results. To kind of give you a little bit further an example or a visual of what invalid or rejected results may look like. This rejected result is that all subsites in the exposure series show an erythemal response. So you can see that we had our test site, we had 5 subsites within it. However, we're seeing erythemal responses at all 5. What this is saying to us is that, 1, the product is not protecting at the target SPF value that we had assumed. It's obviously lower than that. Additionally, we don't know how much lower of a UV dose we could have given to have an erythemal response prior to subsite 1. So because of that, because there is this unknown, this is where we would say that the SPF value is less than or equal to whatever that first subsite is. It's just because that we don't know exactly where the SPF value would have fallen, assuming we would have given even lower UV exposures. In cases like this, we normally suggest 2 ways of moving forward. Either we can lower the target SPF value. That will allow us to give exposures at a lower UV dose, and then ideally we would see some erythemal responses in that lower target. Or at the the client may have the option of just reformulating the product, ideally increasing the active ingredients for SPF and maybe retesting at the target value. Another example of invalid or rejected result is that the exposure series on a subject may fail to elicit any erythemal response. So let's assume this person was exposed, had the product applied, exposed to UV, went home, came back 24 hours later, and there were no erythemal responses in any of the subsites. This is telling us that the product is protecting at even a higher SPF value than we were originally targeting. So again, we don't know if we were to provide higher doses than those that were given, at what point an erythemal response would have shown. In this situation, our suggestion is to increase the target SPF value. That would allow us, again, to increase our doses and ideally obtain an erythemal response. So that is generally the SPF label claim determination test. It's, again, very similar between the FDA and international testing standards, given some minor fluctuations that we went over. The next test that we'll touch on is our UVA protection factor testing. This is another international standard test, which is very comparable to the SPF determination test that we just went over. However, the SPF test utilizes both UVA and UVB of the solar spectrum, whereas this test only utilizes the UVA portion. This test is widely accepted in many countries. However, it's not an accepted method in the US. Instead of assessing erythemal responses or reddening responses, this test uses what's called persistent pigment darkening, or essentially tanning. This test is not required to be performed in most countries. And we say most because some Asian countries actually do require this test for labeling claims. If you use this test, you don't often see a product that's labeled, oh, UVA protection factor X. However, if you do see sunscreen products that have a PA or UVA protection grade claim, this is the test that is used. So in order to get a PA claim, it again is broken down into various levels. A UVA PF of 2 to less than 8 will give you your PA plus claim, and it goes all the way up to PA 4 plus, which is a UVA protection factor of 16 or higher. Like I said, this test is very similar to the SPF label claim for the international standard test. The major difference is obviously one will give you an SPF label claim versus a UVA protection factor claim. The SPF test itself uses UVA plus UVB exposures, whereas the ICE International Standard 24442 test is filtered to only give UVA doses. The subject selection skin type is a little bit different between the two tests. The SPF test requires Fitzpatrick skin types 1, 2, and 3. Again, this is because we're looking for an erythemal response, whereas the International Standard UVA test requires Fitzpatrick skin types 2, 3, and 4, so just a little bit darker of skin tones. The reason behind this is that for the International UVA test, we're looking for a tanning response as opposed to erythemal. Individuals who are skin types 2, 3, and 4 will most likely produce a tanning response to UVA, whereas skin types 1, 2, and 3 tend, or more so 1, tend to not tan. They'll just strictly burn, and then the burn will fade. Additionally, we do use sunscreen standards in all the testing. The international standard for UVA testing just simply uses a different type of sunscreen standard. Then we'll go further into in vitro testing for broad spectrum claims. The in vitro test is not done on living individuals. It is done on what's called PMMA plates or polymethyl methacrylate. Basically, that is just a type of acrylic or a plexiglass plate. There are two major testing methodologies, one again for the FDA and another international testing methodology. Unlike the SPF determination test, the in vitro testing for broad spectrum claim varies more widely between FDA and the international testing methods. So they vary a lot more. There's a lot less in common between these two tests. Both of these tests require the use of a spectrophotometer and a pre-irradiation solar simulator to test the plates. The equipment itself looks something similar to this. On the left, we have our solar simulator or pre-irradiation. It can irradiate up to 4 PMMA plates simultaneously. This solar simulator is essentially a larger version of our single port solar simulator. The opening for the UV exposure is just basically larger in order to irradiate multiple plates at a time. The spectrophotometer you see on the right scans the plates over the entire UV spectrum of 290 to 400 nanometers. It's an automated system that scans the PMMA plates in multiple locations. So each plate will be tested in multiple locations. And then you can store the data in various number of wavelengths, either 1 nanometer, 2 nanometers, 5 nanometers. For FDA specifically, the product is applied at 0.75 milligrams per centimeter squared to the rough inside of 3 PMMA plates. So we do 3 tests for the FDA. The product is spread evenly over the plate using a gloved finger. It requires very light spreading for about 30 seconds, followed by spreading with a bit greater pressure for approximately 30 seconds. Following the application, the plates are allowed to equilibrate for about 15 minutes in the dark. The plates are then pre-irradiated using the solar simulator using 4 MEDs. It's just another UV dose. So it's just another minimal erythemal dose. The plates are pre-irradiated to account for a lack of photostability. Following pre-irradiation, the plates are then placed under the spectrophotometer. The transmittant values are measured at 1 nanometer intervals. On each plate, the transmittance is performed on 5 different locations. And then a control plate is always used. It's coated with 15 microliters of glycerin, so no sunscreen product. In order to claim broad spectrum on a sunscreen product in the US, the critical wavelength of the product must be greater than or equal to 370 nanometers. For the international testing regulation, 1.3 milligrams of centimeter squared product is applied, so a little bit more than the FDA. They use 4 PMMA plates as opposed to 3. The product is spread evenly over the plate using a non-gloved finger. Prior to spreading, the fingertip is dipped in the test product and then wiped to remove excess. So it's essentially you have a little bit of a sunscreen on the finger prior to the spreading on the PMMA plate. Quick spreading is done for less than 30 seconds using small circular motions with minimal pressure, followed by an alternating horizontal and vertical strokes, but with increased moderate pressure for about 20 to 30 seconds, and then this ensures that the entire plate is covered. The plates are allowed to dry for at least 30 minutes in the dark, and the temperature of that dry time should be the same that's experienced under UV exposure conditions. So whatever temperature is going to be under exposure is the temperature in the dark that the PMMA plate is allowed to dry. Following the dry time, PMMA plates are placed in the spectrophotometer prior to any UV irradiation. So if you think about the FDA, they pre-irradiate the plates and put them immediately in the spectrophotometer for analysis. The international standard requires that you do a spectrophotometer reading before irradiation and then irradiated using mathematical adjustment that's given. So the spectrophotometer does a pre or initial reading. It will then dictate how long to expose each plate under the solar simulator. Following that irradiation, the PMMA plates are placed again under the spectrophotometer. So we do a pre-irradiated PMMA plate read and then a post-irradiated plate read. This method is controlled by the use of a reference sunscreen. So the same procedure is followed using this reference sunscreen. So the same application amount, the same method of application, the dry time, the pre-irradiated, post-irradiated exposures. The test is considered validated if the reference sunscreen falls within accepted limits for the test. The International Standard Test also requires a more thorough and regular calibration of the spectrophotometer and a plate transmission test. So prior to running these tests on a regular basis, we validate our spectrophotometer for wavelength accuracy, linearity, dynamic absorbance. Then we also test the plates themselves, the PMMA plates, to ensure that they have the proper transmission so that they are not affecting the transmission of the product itself. For the International Testing Standard, in order to claim broad spectrum, the critical wavelength must be greater than or 370 nanometers, similar to FDA. However, they also have to have a UVA protection factor greater than 1/3 of the SPF label claim. So the major difference between the International Testing Standard and the FDA is that there are more strict criteria that the sunscreen has to meet in order to claim broad spectrum. Just a quick image of a PMMA plate in product application. So product is applied to the PMMA plate using a lot of small dots, similar to the picture earlier that you saw on a subject's back. The product is then spread over the PMMA plate using the small circular motions. The plate is rotated throughout the application to ensure that the entire plate is being covered. And following the circular motions, again, it is horizontal and vertical movements with the rotation of the plate to ensure even coverage across the plate. We also perform some what we call sunscreen safety tests. So sunscreen safety tests can be either a short-term test, which we'll call phototoxicity, or a more long-term test of photoallergy. So the phototoxicity test evaluates the potential for a phototoxic or a short-term response to sunscreen products. The light source that's used for phototoxicity it's a phototherapy unit fit with 4 UVA lamps and a dose controller. It almost looks like a little tanning bed. The test site for a phototoxicity test is the inner forearms. So we use both forearms. One forearm is designated as an irradiated or a UV exposed arm, while the other is left non-irradiated or non-exposed. We have 4 test sites, 2 on each arm. We'll initially tape strip the test sites 3 times, and that's just to remove any sort of dead skin, superficial layers. On the irradiated arm, one site has sunscreen product applied, while the other is left with no product applied. The test sites, or that one arm, is then irradiated with UV. Following the irradiation, a patch with additional sunscreen is put on the treated or sunscreen applied site. Another patch with no product or blank is placed on the untreated site. The non-irradiated arm, the other forearm with the 2 sites, is simply patched with test product on one site and a blank patch on the other. So it's treated in the same manner as the other site, minus the UV exposure. These patches are worn for 24 hours. Following the 24 hours, we remove the patches, and then we visually evaluate the arms at 48 hours and 1 week. A phototoxic response would appear as a severe sunburn. So a phototoxic response, because it would appear as a severe sunburn, is something that we would evaluate on the irradiated arm. The non-irradiated arm is used as a control method to make sure that it's not the product that's causing any issues. It is for sure the irradiation. And the other safety test that we perform is photoallergy. It is a more long-term test, and I apologize, I noticed there's a typo. So photoallergy evaluates for an allergenic response to sunscreen products following multiple UV exposures over an extended period of time. So the phototoxicity test is a short-term response to one exposure to UV, whereas the photoallergy has multiple UV exposures and it's a long-term test. We use our single port solar simulators for this test. The test site is on the back. There are multiple evaluation sites. So we have a sunscreen product that is patched and irradiated, a sunscreen product that is patched but non-irradiated. And then we'll just test the patch materials with no product, non-irradiated, and patch materials with no product, and it will be irradiated. This is to just ensure that it is not the sunscreen itself or the patch materials that are causing any sort of responses. All sites are patched accordingly for a period of about 24 hours. No irradiation if applicable on those non-irradiated sites. 24 hours later, patches are removed. They're wiped with a dry gauze, and irradiated sites are exposed with a specified UV dose, and it's UVB plus UVA, so it is a full spectrum dose. Following the dosing, subjects undergo a 48-hour rest period. After that rest, they return to the testing facility, and we evaluate the test sites for erythemal or irritation responses. The 24-hour patch UV irradiation and 48-hour rest period is repeated for a total of 6 UV exposures over the course of 3 weeks. Following the 6 exposures, subjects have a rest period of about 10 to 14 days, and then they come back for what's called a challenge phase. Subjects come back after that approximate 2-week rest. We patch them again for an additional 24 hours on the same sites using the same patching methods. Following removal of the patches, the test sites are irradiated with UVA only. And then reactions, if any, are evaluated at 48 hours and 72 hours post-irradiation. A product could be considered photoallergenic if we note reactions following this challenge phase. So as mentioned, there are some major differences in SPF testing. However, the SPF test itself, or the UVA plus B test, they differ in some areas, but they're also really similar in others. Some of the most noticeable differences aren't really in the testing methodologies at all, but are actually in the classification, formulation, and labeling of sunscreen products. For example, the FDA classifies sunscreens as drugs, whereas countries that accept international standards may classify sunscreens as cosmetics. Further classifications also exist, such as they would categorize their sunscreens as a low, medium, or high SPF, whereas the FDA doesn't have that type of categorization. FDA and international standards differ in their approved active ingredients for UVB and UVA filters. Currently, the FDA has no strict guideline on the highest SPF value you can claim, whereas international standards limits their claim to SPF 50+. International standards require SPF label claims to correspond with the lowest number in a range of tested results, where FDA allows for any number to be claimed. We'll kind of go into that just a little bit. Label warnings are required, yet they differ between guidelines. Currently, international standards recognizes powders and sprays as accepted dosage forms of sunscreen, where the FDA has yet to include these items in the final monograph. So what's changing in SPF testing? So if you know much about what's going on in the SPF world, things are currently changing and under review. The international standard right now is currently under review with the International Organization for Standardization. It will be replaced with a new standard. It's currently in the completion phase of inquiry as the 13th of June. So the next steps in this process are their approval and publication of the new standard. These expected changes are to standardize the procedure further to reduce potential variations between testing labs. So currently, there are still some areas of ambiguity within the testing procedure itself. So potential changes under discussion are completely removing the Fitzpatrick skin type designation and only using ITA. As we discussed earlier, Fitzpatrick skin type can be subjective, whereas ITA is a quantitative method of classifying skin type. The international standard is looking to clarify descriptions and provide visualization for test product application to take into account varied types of formulations. I believe that they also had mentioned potentially including a video of product application in their regulation. They are potentially doing a new MED scoring system with photographic examples. So you would be able to review their photographic examples and have a clear definition of what a minimal erythemal dose looks like. They are potentially including the use of UV-sensitive film to ensure beam uniformity. They're possibly defining a range of MED determination values. So there would be a specified dose range to give for MED. And then there's also potentially maybe new sunscreen standards with higher SPF values. Those will accommodate when testing higher SPF values of test products. The FDA has published a proposed regulation currently. The goal of this proposed regulation is really to just make sure consumers have access to safe and effective sun care options. So the FDA is really focusing on the consumer and how sunscreen is presented to them. This proposed regulation is expected to, FDA, is expected to issue a final OTC sunscreen monograph to be effective within 5 years or by November 26th of this year. So some highlights of the FDA proposed rule. There are some changes in the proposed generally recognized as safe and effective status of active ingredients. So they propose that zinc oxide and titanium dioxide are proposed as Category 1 ingredients, which means that these ingredients are generally recognized as safe and effective for sunscreen products. They are proposing that aminobenzoic acid and trolamine salicylate are proposed Category 2. So these would be not generally recognized as safe and effective, therefore they would not be usable in sunscreen products. Lastly, the other ingredients or active ingredients for sunscreens indicated, they're pretty much indicating that there's not enough data to support whether or not we can generally recognize these ingredients as safe and effective. So they're proposed as Category 3, so insufficient data is available. They would still be usable within sunscreen products. It's just that they need to do more research to ensure that they're safe. Currently, they are proposing certain dosage forms as considered Category 1 or generally recognized as safe and effective. Many of these are already currently recognized as effective dosage forms: oils, lotions, creams, gels, butters, pastes, ointments, and sticks. Sprays is one that would be fairly new to this monograph. So they would be categorized as safe and effective. Although they would be categorized as Category 1, they would also be subject to additional testing prior to marketing, such as particle size and flammability. Sprays would also have additional labeling requirements as opposed to something like a lotion. Powders will also be eligible. Currently, they're not included in the monograph, but they will be eligible for inclusion in the final monograph. However, they'll be included as Category 3, so basically insufficient data. Powders would also be subject to particle size restrictions. Sunscreens and other dosage forms, such as wipes, towelettes, body washes, shampoos, would all be classified as new drugs and would require a new drug application. Sunscreens and insect repellents, those combinations would be proposed as Category 2, so they would not be generally recognized as safe and effective, and therefore would not be accepted by the FDA. The FDA also proposes a maximum label value of SPF 60+. So no longer would we see SPFs on the market for SPF 70, 100. They would have to be labeled as 60+. There are also some further criteria about maximum testing values for SPF. They would require that a maximum sunscreen be tested for an SPF 80. However, even if it did reach an SPF 80, the label would have to be 60+. They allow for this testing above the maximum label to account for variation in testing. The FDA would also require sunscreens with SPF values of 15 or greater to be labeled with an SPF number corresponding to the lowest number in a range of tested results. So for example, if a sunscreen tested as an SPF 15 to 19, it would be labeled as a 15. SPF that tested as a 40 to 49, so let's say it landed at an SPF 45, it would have to be labeled as an SPF 40. So sunscreens, to add on to that, sunscreens tested at SPF 30 or higher would be labeled in increments of 10. So you would only see SPF 30, 40, 50, or 60 plus. And sunscreens testing at SPF 15 to 29 would be labeled in increments of 5. So you'd see SPF 15, 20, 25. Labeled SPF values according to ranges wouldn't be necessary for any SPFs below 15. So if it were to test, let's say, at an SPF 8, you could claim an SPF 8. The FDA would also require that all sunscreen products with SPF values of 15 or greater satisfy a broad spectrum test. Products with an SPF below 15 would not be required to satisfy broad spectrum requirements. So currently, the FDA does not require any sunscreen to satisfy broad spectrum. However, this proposed change would make that a requirement for SPF values 15 or higher. It also says that they would have to satisfy a revised broad spectrum test. So in order to currently pass the broad spectrum test, the sunscreen just must meet a critical wavelength of greater than or equal to 370 nanometers. That will remain unchanged. However, they also will include a requirement that the UVA1 over UV ratio has to be 0.7 or higher. That is ensuring that the sunscreen has a decent amount of UVA1 protection as opposed to simply keeping the protection in a UVB or a lower UVA range. They're trying to do this because the UVA1 range is more specific to being cancer causing. So they're, again, trying to protect the consumer more than previously. Nice thing about the broad spectrum test on my end is that the current broad spectrum test procedure will remain unchanged. So the procedure that we went through earlier will remain the same. It's just that this new ratio will be calculated using data from the existing test. So all these proposed changes for the FDA are just that, they're proposed. There is no telling right now what will be included in a new final monograph and what will not. So that's just something that we'll have to wait and see when it is complete. Other than that, that is our general overview of sunscreen testing with BioScreen. And then we're just always open to some questions if you have them. You're more than welcome to reach out to myself. We also have Adam, our Director of Sales and Marketing, that is very well knowledgeable in this area also.
Yes.
So thank you very much.
Thank you, Jordan. Yes, we will go ahead and open it up for questions. We did have one come in earlier. Just wanted to remind all the participants, you can go ahead and use your chat box. You can even raise your hand to ask a question directly to Jordan, or you can type it in the Ask a Question box, and we will go ahead and get those questions started. I do want to also remind everyone that we do record these webinars. All participants of this webinar will receive a follow-up email once the recording is available to view. We also post our webinars on our ALS Global website and our YouTube channel. Within YouTube, just search for ALS Limited and then click on the North American playlist. There you will find a wide range of our video resources and also past Webinar Wednesday presentations. So I did want to ask you a question, Jordan. It came from Sophia. She asked, you were around on slide 10 when she asked the question. She asks, how do you ensure that a 2 mg/cm2 amount is applied to the skin?
Sure. So that is where we use our weight by loss method. So to go into that a little further, we weigh our syringe that we use for application, and let's say we're using a finger cup prior to putting product in it. So we'll get a pre-weight. Then we will add product into the syringe, weigh it again, and once the product is then applied to the back, we'll weigh our empty syringe and the finger cup that was used to apply. We'll subtract those weights from one another to ensure that we have the proper amount. So let's say that our test area is 50 centimeters squared, so we have to apply 100 milligrams of product. We'll be able to subtract those amounts and ensure that we actually indeed applied 100 milligrams.
Perfect. Thank you for that response. We do have a few people that were asking just about sharing of the slides and the recording. Just as a reminder, we do record the webinar, the audio and the visual, so you'll be able to watch the entire presentation once it is available. We normally have it posted the following week. So sometime next week, you will receive an email from ALS to let you know that it is ready to view. So you will get to be able to see these slides again. We have, I believe, a possible follow-up question. This is also from Sophia. She asked, can you provide any more details on how the UVA/UVB ratio will be calculated from existing CW test results?
Yeah, so the test itself, because it's measured in 1 nanometer intervals, we get a transmittance measurement at every nanometer throughout the entire spectral wavelength, so from 290 meters to 400 nanometers. And we get that data from every plate that we run and at every location that we run on that plate. So then it is just a matter of calculating the average of the UVA 1 nanometers, dividing it by the total UVA that we collect, and ensuring that the ratio is 0.7 or higher.
Wonderful. Thank you again for that response. I don't see any additional questions. We'll wait just a little bit longer in case someone is still writing their follow-up questions. I do just want to remind everyone that you can follow also our showcase page on LinkedIn. We post links to our future webinars that we will be having, as well as other valuable resources there.